Welcome everyone to this week's edition of the Extracellular Vesicle Club. We have a real treat today because one of the most common sources of EVs and one of the most interesting and important sources of EVs is blood. And ICEV has a blood EV task force, and we're going to be hearing about the latest product from this task force from our lead authors, Fabrice and Dakota. So thanks everybody for joining today. I'd just like to remind everyone to please put your comments and questions in the chat box. I expect there are going to be a lot of them. And we will then allow unmuting at the end of the session. And you will be able to ask those questions. We'll get through as many of them as possible. So Fabrice, I'd like to turn it over to you.
Thanks, Ken. Thanks everyone for joining this journal club. We're very excited, Dakota and I, to take the stage to present this work that has been in the making for almost 2 years, and hopefully this is one initiative among many coming in the future. So I'm assistant professor in the Department of Urology and Immunology at Mayo Clinic in the US. Dakota is medical student as part of the Department of Public Health and Emergency Medicine at Queen's University in Kingston in Canada. And so I will present the first part of the talk, presenting the rationale for developing the MyBlood EV with a couple of evidence-based guidelines, and also Dakota will describe the MyBlood EV checklist, and hopefully with clarity we will promote the adherence to this checklist, and so we can continue to move forward the rigor and the reproducibility in EV research. So Dakota and I decided to present this work at the EV Club, but this work won't be possible without the contribution of many other scientists and clinician scientists. So as lead contributors, Rick Newland, Matt Kalinasi, and Bolly have been, their work have been instrumental to produce this technical note. And as you can see in the front of on the bottom, all the authors who have contributed to produce the MyBlood EV, all those members are part of the ICEV Blood Task Force. So first, a couple of evidence why we decided to develop this MyBlood EV. So as a lot of you may know, rigor and reproducibility is something extremely important in science, and there's many ways to justify why reproducibility matter. The first one that I found very interesting is about the cost. So again, those ones are very like more estimates, but is estimated at about like $115 billion are spent on life science research just in the United States. And there's about 50% of those spendings that are related to preclinical research. However, following multiple studies, surveys among the scientific community, there's a lack of reproducibility that can be estimated to about 50%. So again, when we talk about lack of reproducibility, we mean that the inability to reproduce some data that have been published. And so we can think about, if we have about 50% of irreproducible research, we have spent like about $28 billion coming from private funding, coming from taxpayers, who basically have been wasted somehow to generate the data that are not reproducible. And in terms of what contribute to this lack of reproducibility, there's different categories that have been highlighted. And two of them are very important because we can definitely translate some of those category of irreproducibility into the EV world, such as biological reagents and reference materials, which are seen as one of the major reasons why there is a lack of reproducibility. And something that we found also very interesting is data analysis and reporting. And we're going to go a bit more into detail in the next few slides. Another reason why reproducibility matters is the fact that if we take some of the extracellular vesicle publications in the field of biomarkers that have been published until 2022, we see that there's a lot of publication ongoing on extraphysical and biomarkers, but when we start to look at clinical studies, clinical essays, and commercially available tests, we have a significant drop in terms of the number of publications. Part of the reason is because most of these biomarkers that are discovered, and it doesn't apply only on extravesicals. We can see as a broad field of biomarker research. Most of the biomarkers that are being discovered fail to translate into the clinic. The biomarker pipeline is a very complex process that goes from discovery to verifications to validations, not only in terms of the biomarker per se, but also in terms of the technical validations of the assay that you use to measure the biomarker. And that finish after with the clinical translation. And when there is a successful path throughout those different categories, now we can have a test that could be implemented into the clinic. There's some statistic that was very interesting from the NIH Early Detection Research Network, who actually highlight how most of the biomarkers that are discovered fail to reach the clinic. There's about 56% of all the projects on biomarkers that are in a category that are at the stage of discovery. And more we're progressing towards the pipeline, we realize that the numbers of biomarkers that reach verification, validation, and clinical translation is drastically reduced. Developing biomarkers is extremely burdensome, takes a lot of time, takes a lot of money. It's estimated that about 7 to 10 years is needed to translate a biomarker from the discovery stage to the clinical translation, which means that there's a lot of efforts that needs to be done right at the beginning, at the discovery stage, in terms of reproducibility, so we can maximize the likelihood of translating these biomarkers into the validation phase and potentially into the clinical translation. So what are the factors of irreproducibility in blood EV research? And the first one is definitely the heterogeneity in residual cells, EVs, and also EV-like particles in the blood. So as a lot of you may know, lipoproteins is a major abundant component of the blood and actually outnumber the plasma EVs by more than 7-fold order magnitude. There's also some very interesting work that demonstrated that plasma-derived EVs coming from the tissue represent a very minor subset of the circulating EVs. It was estimated about 0.2% using transcriptomic data, where the blood is mainly enriched in platelet-derived EVs, in B-cell-derived EVs, in T-cell-derived EVs, but the tissue-derived EVs represent a minor component. If we decide to look at the cancer level, but among the plasma EV proteins, less than 0.1% of the plasma EV protein origin from the tumor, which demonstrate that we're really looking at something that is, yes, abundant compared to other sources of liquid biopsies, such as ctDNA or circulating tumor cells. Nonetheless, the tumor-derived extracellular vesicle are very minor compared to lipoproteins or other plasma-derived EVs. And other studies that have been very good at highlighting the heterogeneity in the blood and some of the what we can call confounders that can affect downstream analysis of EVs is one example was published in GTH by the group of Dr. Newland, where residual platelets is commonly found in prepared plasma. So usually we use centrifugations, the single centrifugations, dual centrifugation to eliminate the platelets from the blood and obtain a platelet-free or a platelet-poor plasma. But in that study published 2 years ago, they demonstrated that across different studies and across different samples, we can have a very wide range of residual platelet detected in the plasma, even when we use a double centrifugation, which is now recognized a bit as the gold standard to have a platelet-poor plasma. And interestingly, from the same group, it was shown last year, published in JEV journal, that removing the platelets by filtration and also the erythrocyte ghost can improve the detection of microRNAs that come from the EVs. So in the figures, what we can see is the PCR on the y-axis, the PCR cycle for 3 different microRNAs. And the bar graph demonstrates that the bar on the left, which is just a plasma processed by centrifugation, and on the right, the dash bars is filter plasma. We can see that for 2 microRNAs, there's a significant change in a PCR cycle when we filter the plasma compared to the unfiltered plasma, which demonstrate that depending on how we process the plasma, we could have different results with the downstream analysis. So the pre-analytical variations are now commonly, are now regarded as being a major reason for lack of reproducibility. And to highlight that, there is this beautiful work published by the lab of Dr. Hendricks in Netherlands, so in 2023, when they evaluated the impact of different blood collection tubes on multiple metrics of the quality of blood, such as the extravesical recovery, presence of hemolysis, presence of residual platelets, as well as the EV subtyping using flow cytometry and also some transcriptomics and proteomic analysis. So this study was was rigorously performed because the blood was processed by a centralized lab and was analyzed by some reference lab with expertise in the different techniques. And as you can see on the right is just a heat map to illustrate that depending on the blood collection tube that is used, we can have very distinct results in terms of the amount of residual platelets, presence of hemolysis, and also in terms of the EV signatures that are detected in the plasma isolated, in the plasma of the serum isolated from with those blood collection tubes. So this highlight that it's very important to validate those to compare those different pre-analytical conditions to make sure that we have the most suitable methods to isolate extracellular vesicle. But also we need those comparative studies to really appreciate how variable the data can be when we use like different type of blood collection tubes, centrifugation methods, or again, storage conditions. And another work to illustrate that, so the previous work from Daunt and al. Demonstrated those pre-analytical variations from blood that was processed and analyzed in labs. But we also have this work that is spearheaded by Faize Shekari and Daniel Hagee. So we have been interrogating different biobanks throughout the world. So you can see here, I'm going to show just a snippet of the ongoing work that we have. When we asked about 30 different biobanks in 18 countries, we asked them to provide their protocols to process blood, to collect blood, process the blood, and store the blood, and also what kind of metrics are used to assess the quality control of the samples. On a side note, I would like to mention that this is an ongoing work, and as you can see here, there is an underrepresentation of Central and South America as well as Asia. So for everyone on the call, if you have some contacts, and put us in touch with some investigators in those countries, or even give us contact to some biobanks that can help us to get some information to complete and have a better world representations of the protocol used by biobanks throughout the world, that would be very appreciated. So feel free to reach out to give us some more information. So when we look at the different protocol, and this is again an ongoing work, there's much more information that we hope to present later this year. But we ask some question about like sample collection, transport, type of centrifugation, and quality check. What you can see on the sort of pie chart on the bottom is that different anticoagulants are used to isolate plasma. Different transport intervals, so between blood collection to blood processing, there is different intervals that are used by the biobanks. different centrifugation steps also are used. So as you may know, now it becomes a bit more standard to use 2 centrifugations to eliminate as much as possible platelets from the samples. But as you can see, there's still some biobanks. The majority of biobanks, actually 71%, use 1 centrifugation step, which has been actually the gold standard to eliminate to prepare some platelet for plasma that could be suitable for soluble protein measurement and some more standard analysis. Something that was very, I would say, alarming, but also, eye-opening, was that in terms of the quality check, hemolysis assessment was done visually in about 70% of about 70% of the protocols that we had access to. The platelet count and the lipoprotein count was not part of their workflow. So when we get the samples from the biobank, it becomes very difficult to estimate how much residual platelets could be present in the samples and how much lipoproteins could be present in the samples, which can definitely affect the downstream analysis, especially because we can not only measure, we cannot measure the platelet and lipoprotein count, but also there's no reporting whatsoever which can help us to inform reviewers or against the readership about what kind of samples we have used, so that can enable comparative analysis. And another paper that was also very very interesting and that stimulated or promoted motivations to develop the MyBlood EV was a study from the Spanish EV Society, published in Journal of Extracellular Biology in 2023. When they sent a survey to their members, there was about 70 responses and 43 laboratories accepted to provide a protocol that they use for blood collection, processing, and storage. And they use a codification system that is established by the International Society for Biorepository using the SPREC pre-analytical code. What they found, it was very alarming again, was that 94% of the protocols provided by the 43 laboratories display differences in the biofluid handling. And specifically for the blood, there was no lab with similar protocol. 13 protocols prepare single-spun plasma, 14 protocols prepare twice-spun plasma. And despite EDTA was the major anticoagulant, there was 8 different anticoagulants that was used throughout the different protocols. And more importantly, some labs didn't have access to the pre-analytical condition that were employed for sample preparation. So what is the highlights is that we have an urgent need to establish some transparent reporting of pre-analytical methods, and also transparent reporting of the quality control of the matrix for blood and its derivatives, such as plasma and serum. And so to again, even emphasize what has been presented here, these factors of reproducibility are not limited to extracellular vesicle research. So here is just a technical report published in Nature in 2016, where 1,500 scientists were surveyed to identify what factors can contribute to irreproducible research. And as you can see, the top-rated factor was selective reporting, which means that we don't have a full detailed reporting of, for example, the pre-analytical conditions, the quality of the samples that can help to reproduce exactly the same methods and protocol with subsequent studies. And as part of what factors could boost the reproducibility, how can we overcome those limitations? Interestingly, we need more validations within the labs, between labs, so inter-institutions, and also provide incentives for better practice, formal reproductions, and also having journals that enforce better standards. So, like I said, this is like at the larger scale, right? So this is a picture not only of the EV research, but the whole scientific research. And in terms of liquid biopsy, like I said, we're not alone. So we're very lucky to have individuals as part of the ICEF community that really want to push the initiative of improving the rigor and reproducibility. As you can see here in these publications, there is an international movement with different entities in the liquid biopsy world. This International Liquid Biopsy Standardization Alliance, we actually are working together to establish different initiatives that can help to not only improve, to not only standardize and control pre-analytical variability, but also developing better reference materials and standards, and also having some methods to assess the performance and the clinical evaluations of potential clinical biomarkers. So hopefully, the work that we're doing here is not going to stay in our silo inside the ICEF community, but we definitely want to reach out to those different groups and really embrace the movement so we can, gather forces to improve rigor and reproducibility, not only in EV research, but in liquid biopsy research. And so to highlight a bit like what has been said, in 2019, during the annual meeting of the ICEF, there was a workshop that has been a workshop when the individuals were discussing about how can we improve the rigor and reproducibility in blood EV research. And the outcome of this workshop was published in 2019 in JEV, and those different categories were highlighted. We need a better control of biospecimen and pre-analytical variables. We need to identify evidence-based identification and evidence-based optimization of key variables. And importantly, everyone agreed that we need some sample quality control procedures. And once we know the quality of the samples, we can have downstream applications. And when the study is finally conducted, we can have transparent reporting to help other individuals to maybe not reproduce necessarily the study, but at least having key elements to compare their data with like similar studies. So that was in 2019, and after a couple of discussions with some ICEF Blood Task Force members, there was a couple of us that decided to brainstorm under the wisdom of Rick Newland, who hammer ahead by saying, we need to control the amount of platelets. We need to make sure that we count how many platelets are residual in our samples. So how do we develop, how can we address those different elements in a roadmap and kind of create a new initiative that could be easily implemented by the community? And that's where we came up with sort of three foundational questions. The first one was, what are the critical confounders in plasma serum samples in blood EV research? What are the qualitative and quantitative methods that are used to assess the presence of those confounders? And last thing, what are the recommended strategies that we can use to report the quality of those samples? And so this is kind of like the workflow that we did to produce the MyBlood EVs. So based on those three foundational questions, we developed some surveys. Those surveys were sent first in the ICEF Blood Task Force community, about 16 members. And another survey was sent with similar questions to the whole broad ICEF community, and we had 176 respondents. We get those surveys back, we analyze the data, and based on the results, we developed the MyBlood EVs that was the foundation for the manuscript that was published in JEV. The manuscript was reviewed by 15 members of the Blood Task Force, and also has been submitted to the ICEF Executive Committee to kind of have their approval for the publication of this technical note. And after it has been, again, peer-reviewed and published in JEV. And as you can see, this work has been initiated in July 2021 via Zoom with all those members. And we finally got the publication in December 2023. And so, to build the MyBlood EV checklist, I'm just going to show a couple questions, those foundational questions, and what kind of answer we had, not only from the task force, but also from the broad ICEF community. All those informations are accessible in the supplementary materials of the manuscript. So first question is, what are the levels of concern for the EV-associated confounders? So as you can see on the left, there is a gradient of colors going from extreme level of concern, moderate, somewhat, slight, and none. As you can see with like the dark gray bar and the blue, for every component that was mentioned, erythrocyte components, platelet-derived DVs, non-vesicular extracellular RNA, and non-vesicular protein, lipoproteins, there was kind of like a majority of individuals at the task force that agree that we have a moderate or extreme concern about the presence of those confounders. And somehow it was replicated by the ICEF community. This is very important because we want to make sure that the task force taking those initiatives are not, are actually representing the community and understand really like the limitations that are associated to the blood EV research. So it was very nice to have those two independent surveys and see that we have a very good match in terms of the responses. The second question was, what specific confounders should be reported? And it's not a surprise, but lipoproteins is definitely a major abundant abundant confounders with extracellular vesicles, and the levels, the levels of lipoproteins should be reported, and it was kind of consensus among the ICEF community and the blood task force, as well as the presence of erythrocyte components, platelets, non-vesicular extracellular RNA, and soluble proteins. And lastly, so when we look about how to report the confounders, there's different initiatives now that are available. For example, the MySafe guidelines will propose different different recommendations on how to present, how to report data for extracellular research, not only for blood, but for any kind of biofluid or conditioned media. There's also the SPREC initiative, which actually has been established by the International Society for Biorepository. And there's also the EV-Track platform that some of you may know, which has been established a few years ago and enable investigators to upload in a database the different methods they use to isolate extracellular vesicle from various biofluid and conditioned media. And we also come up with the idea of why not producing a standardized report checklist that is really dedicated to address the foundational questions on what confounders needs to be measured, how do we process the blood, and how do we report, the quality control of the samples. And a vast majority agree that the standardized report checklist should be, developed to address those different elements. So I'm going to stop for now, and I'm going to give the stage to Dakota, who's going to present the different components of the MyBlood EV checklist, and also some initiative for not only the implementation of the checklist, but also some future directions that we thought about, but also the task force has been thinking to kind of continue improve the rigor and the reproducibility. Thank you.
Perfect. Thank you, Fabrice, for that wonderful introduction. So if we just want to go to the next slide there, I'll let you take charge of that. Perfect. So over the kind of last 10 minutes here, I'll just go over the more technical details relating to the checklist itself, as well as the subcomponents of what we kind of came to a conclusion of would be important to include in something like this. I just really want to mention upfront that one of the major considerations to the design of this checklist was to reduce the burden on researchers. We understand going through things like this can be fairly time-consuming, as well as many researchers don't have access to the methods or equipment that would be necessary to get a lot of this information. So keep in mind, one, this was meant to try and reduce the burden on researchers for reporting, and two, these are just recommendations. We understand that researchers might not have all the equipment available, but it's just a form to really standardize reporting and start to get at that reproducibility issue. You just want to hit the next slide. Perfect. So standard amongst many different formats for checklists or standardized reporting, we have some very essential basic study information. I'm not going to go through it in great detail, but these include, of course, the manuscript title, corresponding author, institute, the specific cargo that your research is looking at, as well as the source of specimens and years of collection. So that relates specifically to when samples were collected and subsequently deposited either in a biobank or utilized for experimental means. Up next was very standard blood collection and processing information. This information should be readily available by most phlebotomy clinics, but again, we understand that this information might not be available through biobanks. So if the information is there, it's wonderful to put in, but of course, if the information is not there, don't worry about it. It is essentially a checklist, just highlighting details that would be great to report for studies to better understand the reproducibility. So a few key details here, of course, patient fasting status, the length of that, anatomical access site, specifically blood collection tubes and which tubes are being utilized. I think Fabrice really nicely mentioned that there's such dynamic protocols in use currently, and that includes both amongst individual researchers and biobanks. So just having this information on hand is great to look back and allow others to understand why their results might be different between studies and really start to get at that. We have other things here: transport temperature, centrifuge branded model, bucket rotor, the number of centrifugations and the speed associated with those, as well as the storage tubes and the storage length of time. Following up with this, we have a number of different plasma and serum quality control metrics that were built into this, and this was largely based off feedback from both the ICEV community as well as the task force itself. In mind, we're really trying to minimize the number of variables that were included in this and include only essential things that would really help better understand how a study was conducted and what the results might mean. So these include the number of freeze-thaw cycles, the thawing temperature, different metrics related to hemolysis, whether this was done visually or through a more standardized clinical lab device, and the number of hemolyzed samples that were included as well as discarded. More so, we're including different metrics on platelets. And of course, we just want to know, were the presence of platelets detected? What method was used? Was it through flow cytometry? What was the concentration? How did you measure these? And of course, this will be a dynamic process. I want to reemphasize later on that this is version 1. This will be revised throughout time, throughout different feedback, and will hopefully be made more applicable and simpler for researchers. Following this up, we have lipoproteins, which again were one of the most recommended reporting metrics for EV studies. Whether or not lipoproteins were detected, what kind of lipoproteins were detected, and how they were measured. Again, very standard metrics for assessment of study quality. So this was really the generalized reporting guideline we were working off of. We really wanted to start addressing this transparent reporting crisis in EV research and tried to simplify it. So the way we're currently envisioning reporting through this checklist is downloading the MyBlood EV table, either on ICEV or through the online manuscript, completing and attaching to your manuscript submission, the supplementary materials, this checklist, filled out as complete as possible. Uploading the MyBlood EV table to an online database, which again is linked through the manuscript. And then through kind of a dynamic evolution process, we hope in the future that we can conduct meta-analyses and validation studies off the checklist uploaded. So this would include a comparison, a head-to-head comparison of protocols for downstream applications to better assess which protocols might affect things like lipoproteins, platelets, or other key variables. And then through that, develop guidelines and recommendations that are evidence-based instead of purely just expert consensus. So again, taking this data, assessing what it means in context of the results, revising the MyBlood EV checklist, and having actual evidence-based guidelines for the future. In terms of our short-term goals, we really want to disseminate this to other international societies. We're not working alone. I think, as nicely mentioned, there's a lot of different societies currently looking at standardized reporting, whether it be through ctDNA, other bioliquid societies, ISTH specifically. So disseminating this to other societies so that we can better understand their needs and they can better understand our needs in regards to biospecimen utilization. Adopting this EV framework by public and private biorepositories will be a next major step. Having them on board with standardizing things like centrifugation cycles, transport time, or even just having the information available for researchers will be essential in the future. And then moving along a little more, having a template for things like myUrinEV or MissEv-EV, just expanding off what we already know and integrating this framework into other biofluids. And then looking to the future, we really want to develop this virtual EV biorepository, or VBIR, all-in-one interface. So this is essentially a grand scheme interface that the research community could use for better understanding study results and gaining a more holistic understanding of how results fit into a bigger picture. It would really start with developing the standardized framework from MyBlood-EV for collection, handling, and storage, looking at specific quality controls and what that means in the context of results, understanding how MissEv fits into this for EV isolation and purification protocols, and then using something like EV-Track to integrate proteomics results, RNA results, as well as single EV analyses, and integrating that data into a single server. So essentially having a standardized reporting framework that we can then analyze and take a look at both the processed EV data as well as clinopathological data, and use this process as an iterative framework. So take all that data, look back and see what it means in regards to study results, and potentially use that, number one, as a confounding adjustment, but also number two, as I mentioned, to develop more evidence-informed methods going forward. And of course, there's a number of different things at play here. So we need to really understand how EV biobanks, or biobanks in general, could integrate this standardized framework, as well as how the research community can better utilize this framework and how we can further adopt it nationally or internationally. Final slide. So I think at that point, we're happy to take any questions and answers, or answer some questions. So thank you very much.
Thanks, Dakota. That was very good. Yeah, so if you guys have any questions, please feel free to use the chat, or you can also and we give you the opportunity to turn on your mic and ask your question. There's Dolores who mentioned, like, in a biobank presentation, I did for the biobanks, I mentioned that only 7% of them were doing platelet counts and 3% lipoprotein assessment. So the specific method to assess both. So I believe the platelet count was done using hematology analyzer, and the 3% and the lipoprotein assessment was actually for a specific biobank that was specifically designed for a clinical study, and they were using spectrophotometry to measuring those lipoprotein with the what they call the L index, which is absorbance value associated to the lipoproteins. You can unmute yourself also.
Yeah, thank you. So basically, the platelet count was not done by flow cytometry using CD41 and CD61, as you mentioned later. Okay, that's interesting.
But this is interesting because, I know Rinka has mentioned that in the past, that, sometimes the residual platelet count could be under the limit of detection of some of the hematology analyzer. That's where, using flow cytometry, conventional or even high resolution flow cytometry, can really help to get a true picture of the amount of platelets that could contaminate the sample.
Yeah, and also, can you elaborate a little bit on the lipoproteins and how specific is the spectrophotometry you said?
Yeah, so I mean, the measurement is really, so when we build, we look at methods. When we build the MyBlood-EV, we look at methods that could be used to assess potential lipoprotein. I don't want to say contamination, because it's going to be in almost every sample, but the lipoprotein presence. So there's something that is more sorry if I don't pronounce correctly, turbidimetry. So basically, it's like a sort of like the visual assessment of presence of lipoproteins. When you exceed a certain amount, the plasma is not going to be very clear, it's going to be turbid. The spectrophotometry is really like measuring an absorbance that is more associated to the lipids, so the lipids content. So this is kind of one of the methods that, I mean, it's not widely used, but this is an index that people understand, for measuring lipoproteins. And otherwise, after, there is the methods like ELISA, Western blot, which require, like, for example, ApoB antibody or ApoE antibody. So, I mean, this is where we, like, we propose those methods, but we understand that, depending on the size of the cohort, you cannot do on every sample. So that's why, if you have a large cohort, but you want to prove that, yes, we looked at in our cohort, lipoproteins, is present, but is not like at the concentration that definitely can have a negative effect on the downstream results, meaning that, we don't know yet the range, but there's some sample that can present abnormally elevated amount of lipoproteins. So for that, you can use a fraction of the samples of your cohort and either use spectrophotometry, ELISA, or again, Western blot. I mean, we put it out there. That's going to be very interesting over the years to see how people, report those informations. We understand that it could be complex. We're doing ourselves the same exercise, so that's going to be interesting. But we wanted to put some techniques that are available to everyone and doesn't require much reagents or tools to measure it.
That's great. Can I ask a very quick more question?
Go ahead.
Do you know if anybody has used CD41 or CD61 functionalized beads to remove the platelets? And with what result? I mean, does that work?
Yeah, I don't know about that.
Right.
That's
I'm not sure either.
Yeah, I don't know. That's a good question,
Okay.
Boris.
Yeah, I think it would be nice to know. I mean, lipoproteins are really overwhelming, but if those two major entities could be depleted, I think that would be good.
Yeah, that's an excellent point. Absolutely. Dakota, I'm going to read the question and I'm going to let you answer this one, okay?
Sure. Yeah.
So this is a question from Keith McRae. I noticed that in our analysis, there was no use of gel filtration, for example, QF columns in EV isolation. Is this not felt to be of value?
So I hope I know what you're getting at there, Keith. We focused mainly on the pre-analytical processes here. So of course, gel filtration, I think personally, is of great use. There's a number of successful methods that have been used to kind of sub-analyze EVs from plasma based off gel filtration. But in terms of what we were looking for in this framework, it was all pre-analytical. And we kind of left the post-analytical stuff to MySev and their suggestions for quality assessment or EV isolation. Is that kind of what you're getting at there, or can I clarify something?
Yeah, no, thank you. Yeah, I think pretty much. I had thought that there's some evidence that is helpful in separating out EV from lipoproteins, etc., even on a pre-analytical basis, but haven't seen as much on it in the last while as there were a few years ago.
Yeah, no, for sure. We try to keep most things just purely before the plasma or as the plasma is generated, and then once EVs are separated out, left that to a different task, essentially.
Okay, got
it.
And
I believe, with the updated version of the EV track that I know is in ongoing, that would include some of those metrics for post plasma and serum isolations, and going more down the road using, like, the different techniques to kind of like enrich or isolate extra vesicles.
Okay, thank you.
Hernando Del Portillo put a very interesting questions. I'm gonna ask you to unmute yourself if you wanna ask questions.
I have done it. First of all, thank you so much, Fabrice and Dakota, for such an excellent and motivating talk. I'm a little bit worried about the fact that, there are, low and middle income countries where this standardization in blood banks will be very difficult as long as the blood is good for, transfusion. And most important is the fact that many of the samples that we take in these infectious diseases of poverty are taken in very low income, very low isolated areas where it's very difficult to get. So the checklist, I think, is an important issue, but I think we have to take into consideration that for this kind of settings, this checklist somehow should not be used as an, a way that the paper should not be reinforced. So I would like to know your opinion about that.
I'm happy to take a first stab at this one.
Go ahead, Dr.
I think that's a very valid point, and it's one we definitely considered as we were developing this and writing the manuscript. The last thing we wanted was to feel like researchers had to do this. That wasn't the intention at all. We wanted to have a tool and a resource available for people to say, hey, I have one or two of these variables that I could plug into this checklist to maybe make reproducibility or make it a little bit more transparent rather than having this big kind of cloud of materials and methods of what to put in there. But I completely agree with you. There's certain situations when researchers aren't going to have the resources available to complete this. And we really tried to make sure the wording in the manuscript emphasized that it's okay if parts of this are completed or if all of it's not completed. It's merely just a template for researchers to better understand how to maybe make their study a little bit more rigorous if it is available. Because you're right, there's many situations in countries where the tools aren't going to be available to do this.
I thank you, Dakota, because I remember with the previous checklist, you had like a minimal percentage that you have to accomplish. And many times we were not able to reach to that percentage. So this is very important to bring to the community. Thanks a lot. It was highly motivating. Okay. Thanks a lot to both of you.
Thank you. Appreciate it.
Bye.
We have a question from Patricia who asked, what about using protease inhibitors after serum collection? Do you recommend this procedure? So I'm not going to make any recommendation myself. I don't know, Dakota, if you want. But I don't know if we have, I don't recall any studies who really looked at the impact of protease inhibitors, adding to the serum after collection. So I won't have an answer, but what I can say is that this kind of MyBlood-EV checklist is a beautiful support to actually test such a comparative study. So looking at having some bloods processed in a uniform manner and maybe having a comparison of adding protease inhibitors, no protease inhibitors, and see how it could impact some downstream analysis. So I don't have an answer for that particular questions, but the whole initiative can help to support to answer that specific question. I'm gonna ask Mega to unmute to ask questions that was posted on the chat.
Yeah, I wanted to ask that sometimes, like I have observed in my own lab, sometimes some samples are hemolyzed and some are not, and some samples are processed immediately, and some have to wait for a while. Then what happens in such a situation? Do you have records of such discrepancies in following the standard protocols within the same lab?
So are you referring about what to do when samples have been processed differently inside the same lab?
Yeah.
I mean, we don't make any recommendation about what to do or what not to do with the samples. This could just reflect also a situation that is experienced by multiple other labs. So I would say that I mean, from my perspective, every samples at certain level can be used for EV research, right? We understand that the samples are precious and we don't necessarily have access to larger batch of samples to EV research. So the only suggestion I would do is that just to report, so if you have multiple protocols that have been used to process the samples that you present in our study. Let's say that you have 3 different protocols, but you can report 3 different MyBlood-EV checklists. This is just one per protocol, and is really to help the readers and also other studies who find your data interesting. Okay, how can I compare my data with your study? So if you have a transparent reporting of those different protocols, we can have a better understanding on how to compare apples with apples. So that's the only recommendation that I would suggest. But obviously, as much as possible, there's no there's no gold standard right now. We cannot call that there is a gold standard in terms of how to process blood for extracervical research. With the work that the lab of Dr. Hendricks has done with the different blood collection tubes, this is the kind of work that we hope we can see for multiple preanalytical considerations, like number of centrifuge, speed of centrifuge, temperature. So this is the work that is not necessarily elegant in terms of how does he help to make major discovery, but we need those studies to, after a couple of years, we can establish what Dakota described, some evidence-based guidelines where we know, for example, that, okay, one centrifugations, we know that this is not enough, and we can make that as like a consensus statement. So everyone is moving toward maybe a double centrifugation. So I was taking just as an example. So that would be my recommendation.
Completely agree. I think we have a question as well from Christina here.
Yeah, Christina.
Hi there. Wonderful talk. Thank you. I just have a question. You've answered some of them actually just in your discussion. Is this something that we need to attach to all papers going forward? So any manuscripts that we're submitting, you would like to see this form filled in and attached going forward? And then for people, we've always done 2 centrifugations, but if people have different, if we get cohort samples from other people and they've just done 1 centrifugation and obviously now it's been frozen, so it's not going to be the same as what we've processed. Can we only compare within that cohort, or can we compare to our cohort? How are we going to address those types of concerns? Because obviously if we're collaborating with people and they do slightly different things ahead of time, maybe 30 years ago when they collected the blood, how can we compare?
Do you want to take a hit, Dakota?
Sure, I'll take a first pass at this one. So regarding your first question, I think it would be great if people could fill this out and submit it with manuscripts. Of course, there's no complete requirement for it, but we're just suggesting to researchers that it would be great if people could fill this out, of course, to the best of their ability, understanding that there is parts people can't fill out just because of equipment limitations or other limitations, but it would be great to have this checklist available so that we can do meta-analyses in the future and develop those evidence-based guidelines. About your second question, yeah, it's always tough to look back and figure out what exactly to do with this sample in the freezer that's been there for 15 years and has had 1 centrifugation cycle and very little information. So I can definitely relate to that. My response would be to this, and Fabrice, you can kind of add on to this, would be potentially filling out the checklist as is and just adding a comment saying X number of samples had this done, X number of samples had that done. And unless we have the very detailed kind of metadata from each experiment, I personally don't think it's going to be easy to deconfound that, but just having that very surface level data available might help us better understand results in the grand context of EV research.
Yeah,
That's great. Thank you. Yeah, because it is interesting that you commented something like 71% of people do only do that 1 centrifugation, and we collaborate with a lot of people, so that is more normal than what we do.
Oh,
so
yeah.
It's interesting.
I mean, this is a very good point, and also, it's always so the difficulty when the challenge when we developed the MyBloodEV was to be very inclusive, right? That it could apply to everyone. So it's also going to come to what downstream application you have. So for example, like, our lab do a lot of flow cytometry, and to be honest, like, do I care that I have a lot of platelet-derived EVs when I measured, I don't know, another type of EVs, like tissue-derived EVs? Maybe not. Yes, maybe they're present in my samples, but I don't mind because I use a single vesicle analysis and I'm chasing, like, a tissue-derived EVs. Now, if you start talking, which is very popular, like transcriptomics, like mRNA detection, proteomics, all those kind of like more profiling technologies, you isolate the extracellular vesicles using different kind of methods, centrifugations, SEC, combinations. There's no perfect method that's going to remove everything that you don't want, right? So the thing is, if you have some residual platelets, and you do like, proteomics or transcriptomics, whatever you measured, you need to take into consideration that some part can come from those platelets, right? So the idea is that, oh, I found these amazing signatures of microRNAs, blah, blah, blah. Okay, fine, but I have no idea what your samples was made of, right? But if you say like, oh yeah, I found these signatures, but I estimate that I have maybe, I have X amount of platelets or like 5%, residual platelets. Okay, good. At least I have a reference. At least I have something that tells me, okay, I can appreciate, what kind of sample you work with. Not saying necessarily, oh, this is bad, but just, having informations that we can use, that if we ended up by having exactly the same conditions, can I replicate this kind of result? So that's why, we kept with those three major confounders: lipoprotein, hemolysis, and platelets. And hopefully, moving forward throughout the years, maybe we're going to have a way which is going to be supported by the methodology we develop to say, okay, now we can have a component of measuring, I don't know, platelet-derived EVs, because, they're very abundant. And maybe we have, like Dolores mentioned, maybe we're going to have a way to remove them for people who don't work on platelet-derived EVs, their own contamination. So we see how the field progressed. This was very like a first version, but what we hope is, we don't want to discriminate any studies. What we want to know through this checklist is, one, the adherence. Can people are able to complete the study? If in 5 years down the road we see that there's like no one is attaching that MyBlood EV checklist to the manuscript, but we're gonna say like it's not because the community is bad. There's maybe something that is not feasible or difficult, and we need to rethink the project. But I think the format that he is very fast to complete. People put as many information as possible, and in a couple of years we'll be able to present that checklist and say, look, this is what we realized, and how do we move forward?
Fantastic. Thank you so much for all your work.
Thank you. So I'm going to go quickly on a couple of questions. So Augustine was asking, did you found the correlations between the abundance of certain particles, such as lipoproteins, and a potential to confound research outcomes? In this sense, could the concentration of particle in blood plasma provide an indication as to what to pay more attention in terms of confounding, or what to prioritize for depletion? Yeah, that's a very good question. I mean, I have some correlations in my own studies of the presence of lipoproteins when you do proteomic studies. I won't go too much in detail, but I think, especially the very abundant, and I always refer to those transcriptomic proteomics. Funny thing, I've been working with a couple colleagues who sent me the proteomic data from different kind of disease, cancer, no cancer, healthy donor, who like basically using platelet poor plasma, doing proteomics. And despite the methods that was used to isolate the EVs, there's always, I would say, 50-60% of the protein content that always come from the same proteins. Like, there's something that is inherently present that we always see, like such as like the hypolipoproteins, or again, like some coagulation factors, for example. So now, how do we develop technologies to improve the depletion of those materials? I leave the door open. I think there's some work ongoing right now, and hopefully we have some studies ongoing. I'm sure Ring joined the call, so I guess like for example, like the techniques to remove the platelets, like using filtration or the type of method Dolores mentioned, immunocapture. So I think like using this checklist, maybe we're gonna see more optimization studies that could help improve the quality of the samples for downstream application.
Yeah, many thanks for this.
Just to add on to that, I think it's absolutely valid to think down the line, once we have enough data, that we could better understand the potential correlations, right? So I think I'd be amiss to see Rank smirking there, and I'm sure it's about platelets in plasma, but just knowing that tons of platelets are there, and in my own studies, at least for transcriptomics and things like that, we know there's a massive contribution of platelets to the signature we're looking at. So down the line, could we have enough data to understand, hey, one or two of these markers is maybe more platelet EV associated rather than the specific indication we're looking for. And I think that maybe is a long-term goal for this project.
Many thanks. Yeah, great talk. Thank you.
Thank you. And so we went through like most of the questions. And again, thanks a lot, everyone, for your nice comments. We really appreciate the support of the community in this work. We hope as a community that people will take seriously this initiative and try as much as possible to complete the MyBlood EV. As you will see in the manuscript, we ask you to upload in a very simple shared drive that we created. This is the first step. We want to get all those editable PDF, but after we're going to come with an interface that is going to be more aesthetically appealing, and also going to include other type of quality control checklist. And like Dakota mentioned, having the inclusion of the EV track, the NanoFlow repository, so on. This is a work in progress. So we have some bright days in the future. And we have Dr. Bouzas who raised her hand. So yes, Dr. Bouzas, go ahead.
Yes, thank you, Fabrice and Dakota, for this excellent, really excellent and inspiring presentation. Very important work. I would like to congratulate Ring and the whole Blood EV task force and encourage everyone in this call to carry on paying a lot of attention to all those factors that have been mentioned in this talk. And finally, I would like to thank someone else. I would like to thank Ken Whitwer, on behalf of ICEV and on behalf of the entire extracellular vesicle community for initiating and for running this EV Club for years. As you may know, other responsibilities of Ken seem to take him to other directions, and the ICEV leadership has a very strong will to keep the EV Club alive. This is such an amazing initiative that it would be really a shame to let it die. So until can come back again and take it back from us, we would like to take it over. And as I said, we cannot be more grateful than this to Ken for initiating and running the EV Club for years. Thank you for all these amazing efforts.
Great message, Edith. Thank you very much. And before we close, I'd like to give the last word to Ring, who has been like a mentor in this entire efforts of developing the MyBlood.
Well, I'm not sure what I can add to what you and Dakota have said already, but I thank you very much for this excellent presentation, because I think what also Edith said, I think it is good that we we raise awareness. I think even the fact that just the checklist is there and that people see it already may set people thinking, and I hope that it will help to improve the results we get from our EV research in the future. Thank you. Excellent work. Well done.
All right. On that note, everyone, I think we went through all the questions, and is at least in my time is already an hour past. So thank you very much, everyone. It was wonderful to have you on the call, and we're gonna close this EV Club. And like Dr. Bouzas mentioned, stay tuned for more adventures with the EV Club and more. So thank you very much, and have a wonderful rest of the week.
Thank you, everyone.